I have a wine fridge with thousands of fruit flies in my spare bedroom. Yes, they're alive, and yes, I have a very supportive partner. Fruit flies are known scientifically as Drosophila melanogaster, and they're a very popular model organism in biology. Over 100 years ago, researchers first learned about chromosomes by observing how the genes for white-eyed flies are passed from generation to generation. And now they're used to study genetics, behavior, neuroscience, and disease. Over 75% of human diseases with a genetic basis have an analog in fruit flies. They get cancer, Parkinson's, depression, and more. I don't know how you tell if a fruit fly is depressed. I'm a mechanical engineer, and I first learned about Drosophila with a conversation of a friend many years ago. I was surprised to find out how much manual labor is involved in fly experiments, and eventually I started a company to automate that work. Biologists have identified thousands of mutant strains of flies. For example, there are flies that are missing proteins involved in circadian rhythms, and they don't sleep. But the problem is you can't tell they're mutants just by looking at them. So they've found a way to attach marker genes to these interesting flies. Marker genes can change eye color, wing shape, or even make flies glow in the dark. So biologists have to sort flies to identify genetic variants, and they separate males and females to set up crosses. A researcher could spend hundreds of hours each year just sorting flies. How do they do it? Biologists use carbon dioxide to knock out the flies and pour them out on a pad. And under a microscope, they use a paintbrush to push the flies into piles. It gets tedious. It's 2015. Can't a robot do it? So I founded a company 2 years ago to try and do this. And after a little bit of preliminary work, my brother joined me. He's an expert in computer vision and machine learning. And together we built a robotic fly sorter. It uses a high-resolution camera to take pictures, software to analyze those images and decide whether a fly is, say, male or female, and then sucks it up via tubes to vials. The system is 97% accurate, which is pretty good, right? Well, that translates to 3 mistakes every 100 flies. And those 3 male flies hanging out in the vial with 97 females, they're having a really good time messing up your experiment. When you knock out flies, they fall in a variety of orientations that you just can't predict. And a lot of the uncertainty came from trying to analyze those images. Around the time that we decided to try using flies that were awake, a group in Switzerland published a paper that talked about their system doing exactly that same thing. They built something called the fly catwalk that takes pictures of flies walking in between 2 pieces of plastic. Now, their system is remarkable. It's open source, but it's also very large and very slow. So it only gets 1 fly every 60 seconds or so. They wait for flies to walk into the chamber, and then they close the door behind it. And if there's more than 1, they have to puff them out. It's a little bit like playing biological slot machines. So my brother Matt and I came up with a couple of ideas to try to introduce flies into an imaging chamber like theirs, but more quickly. If the idea worked, then, well, we would have a product, and if not, it'd be time to close up shop. Many 3D prints and laser cuts later, I'm happy to report, one of the ideas did work, and the company is still around. I've spent the last 6 months working on and perfecting a fly dispenser that spits out an individual awake fly every couple of seconds. It uses foam wheels to act as a valve and basically limit flies flowing through a narrow chamber. I've built 50 prototypes, and it doesn't squish flies anymore. It's all good. And it turns out this is useful to behavior researchers on its own. They need to load flies 1 at a time into their experiments. So it's going to be a big 6 months for us. The fly dispenser is in beta. It's going to be on sale soon. And then we hope the automated sorter is close behind. We hope you'll check out our blog at sexmyflies.com and learn more about our work and also the amazing fruit fly. Thank you so much.